We are joined now by Dr. Sumantra Chatterjee with the Johns Hopkins University School of Medicine. Thanks for joining us.
Thank you very much for having me.
You will be co-moderating a session titled Looking Beyond the Genes. Tell us about it.
So, we have reached a stage in human genetics now where we're moving from just sequencing-based studies to a more functional approaches, where we're going back to the labs and asking the question, what does a mutation mean when a patient gets that mutation? So the session was organized with that hope that we can get it to a larger community, that it is important now not only looking at sequences, but sequences in genes, beyond genes, and what the functional consequences of the sequences are. And that we hope will sort of inspire the community, and especially the younger trainees in the field, to sort of look beyond what we're already doing, a new epoch of human genetics, if you can call it.
Why is it important to look beyond the gene?
Because most of the mutations we're finding now are actually beyond the genes. They're not in the genetic actual gene body. And so the important question then is, why are so many mutations not in a gene if it's a genetic disease? So if there are so many mutations beyond a gene, then what do they do? And it's no longer, as I said, it's no longer good enough just saying they're there. It's now understanding why they're there. And again, going forward for any disease, not only the disease I work on, but any disease, what does it actually mean to have that many mutations looking beyond just pure gene sequences?
What specific topics are you most excited about exploring at this session?
So I'm very, very excited about all the speakers in the session, and because they're talking very disparate topics, but as you see in the session, all the disparate topics come together into one coherent story of how now we are have an ability in looking at the genome to understand why certain mutations occur where they occur, understanding those boundaries of genes, understanding why certain pieces of DNA get more mutated than others. And you will see my colleagues working in different areas in this field are working on something called TADs, topologically associated domains. They're looking at enhancer functions, they're making mouse models. So each of these are very, very, very exciting, and we hope going forward will really lead us to a new era of human genetics.
And you yourself will be giving a talk at this session. Tell us about it.
So I work on a specific disease called Hirschsprung disorder, which affects about 1 in 5,000 newborn babies, who are born without no or very little neurons in the gut, or nerve endings in the gut. And it could be lethal if it's in an extensive form. So my work basically looks at something which I said about these non-coding variations which are not in the gene body. And what we have found basically is those mutations on their own explain much more of the disease than what we had in the last 30 years we had found mutations in the gene. So what I'm exploring in the laboratory right now is understanding why those mutations together, what they do, and patients, and sort of use that as a sort of a benchmark for patients who have these various combinations of non-coding mutations, and explain if we can explain the disease better based on what we know of the function of those mutations.
Fascinating. And what do you hope that attendees gain from this session?
Right. We are really hoping, and we're thankful to ASHG for giving us a really good opportunity, as well as a bigger room to present this work today. And we hope that a larger community of human geneticists take away from this that we have now reached that cusp in human genetics where we need to move forward from just sequencing-based studies to actually looking at what the sequences mean and why are they there, what do they do, and we have to go back to the bench and do the hard yak and get to the real hardcore matter of why the disease is formed. And this cannot be done anymore by sequencing. We've done an excellent job of sequencing. Now we need to understand what the sequence mean. And that is what I hope the human genetics community take forward from there, and going forward, especially the younger trainees going forward in this field, really do what we hope we want to do.
The future sounds exciting. Thank you so much, Dr. Sumantra Chatterjee. Thanks for joining us.
Thank you very much.